Our first presentation is on genomics with Dr. Margaret Gulley from University of North Carolina at Chapel Hill.
Well, thank you. It's really a pleasure to directly interact with patients and their families today. As a pathologist, I don't get to do that. Usually in my day-to-day work, I look at samples, specimens, blood and tissue specimens and tumor specimens. But to meet patients face-to-face is really an honor for me, and I'm also really impressed with the advocacy and the education work done by the foundation, so congratulations for that. Today I wanted to tell you a little bit about genomics of gastric cancer because gene testing is a really hot topic. There's more and more gene testing that's coming into routine patient care, so we'll talk about the tests that are used routinely for gastric cancer, and then we'll also talk about some of the latest research that is showing promise for new gene tests that will be informative for treatment selection, for monitoring patients, and even for gastric cancer prevention. So I think there's a lot of opportunities to really build on what's going on in gene testing in the field of gastric cancer research and clinical care. So pathologists are probably the kind of physician who has a high impact on gastric cancer patients, but that you're least likely to actually meet in person. So the pathologist is the person who makes the definitive diagnosis of cancer. And they do this work mainly by looking through microscopes at tumor tissue and not only defining that there's cancer there, but classifying it in a way that helps patients to get the best possible treatment selections. So we'll talk a little bit about what pathologists do and the ultimate expression of what pathologists typically do with a tumor sample is issue a pathology report, as the one shown here, that really does provide the diagnosis and a bunch of other information. Virtually all patients do have access to a copy of their own pathology reports, and so if you're interested in learning more about how to interpret all the complex terminology and wording that's on a report, I would refer you to this website here that has some valuable guidance for patients. In terms of making that diagnosis, looking through the microscope and doing that histopathology evaluation is always part of a tumor tissue workup. But increasingly, pathologists are using a lot of other tools and including looking at past biopsies or tissue specimens from the same patient and comparing them, doing immunostains, which is basically a way of looking at protein expression. But what we're really going to talk about today are the DNA and RNA-based gene tests that are used in gastric cancer workups, and that's what I'm showing you here on this slide is the in situ hybridization assay. I'll show you two types, one for HER2 and one for Epstein-Barr virus, that are important in this disease, and also polymerase chain reaction and sequencing assays for either DNA or RNA, and then methylation-based assays, which is basically a way that that genes get turned on or turned off in cells as another mechanism besides gene mutations for altering which genes are expressed in cells and therefore what those cells are programmed to do. So at the end of all this work then, a pathologist will issue a report to the patient's medical record with their interpretation of the microscopic findings as well as any ancillary tests such as the ones listed on the slide here that were done that really defines, in many cases, either in a small biopsy, the initial diagnosis, or in a larger resection specimen, the extent of the tumor, and other things that are really important in the prognosis and in the selection of therapy. There are a lot of gene tests that are out there now. Very few of them are for gastric cancer. I'll get to those couple in a minute. But this website, which is run by the US government, is a registry of all the gene tests that are out there now. And basically every human gene is now part of testing for one purpose or another. And you can see 11,000 different diseases or medical conditions are now covered with certain gene tests, done in many different testing labs, including academic centers and reference labs, and even community hospital laboratories. You can also see the increase in the number of gene tests over the years. This has been really a revolution in medicine in terms of the availability of gene tests to be used clinically and also to be used in clinical trials, where we're testing new ways to help us better predict which patients need which therapies. And so these gene tests are not just for cancer. I want to emphasize that these gene tests can be used in gastric cancer, but also in different tests are used in different kinds of cancer. And not only that, these tests can be used in infectious disease diagnosis, because every infectious organism has its own genome, for which we now know the sequence of every human pathogen genome, and we can design laboratory tests to diagnose infectious diseases. Infectious disease is actually more important, we think, in gastric cancer than most other kinds of cancer, because there's not one but two infections now linked to gastric cancer. One is a bacteria, Helicobacter pylori, that predisposes patients to get gastric cancer, especially if it has certain virulence alterations in that bacterial genome. And the other is Epstein-Barr virus that I'll show you some more information about in just a minute. So in any case, that's just yet another application of gene testing that comprises some of those many 11,000 different conditions we were talking about. But the next is heritable disease, and we'll talk in a few minutes about some heritable risk factors for gastric cancer that are basically running in certain families. And then finally, identity testing is next, which if you've been watching TV shows, you probably have this impression that that's what pathologists do, is look at evidence from crime scenes and solve crimes. That's not the kind of pathology I do, but it is what the lay public generally thinks of along with autopsies when they think of what pathologists do. But actually, the vast majority of pathologists are doing similar things to what I'm doing, which is looking through microscopes and running clinical laboratories that are responsible for looking at the tissues and the blood samples and all the zillion samples that it takes to actually manage patients with gastric cancer over their course of treatment. And then finally on this list is pharmacogenetics, which is the super hot topic. So when we were talking about targeted therapy and immunotherapy this morning, a lot of that kind of therapy is based on the results of laboratory tests, and some of those are gene tests. And so let's talk about the two gene tests that are used in gastric cancer patient management. And these are the two that are used routinely, and they're part of the practice guidelines to do these tests in certain situations. So the first test is ERB-B2, which is the official genome, but everybody calls it HER2, so we'll call it HER2 today. HER2 gene amplification, which can be found by immunostain, but in terms of the gene tests, it would be a gene amplification or sometimes a mutation of that gene. Basically, HER2 is this receptor that sits on the surface of a cell, and it waits for some signal from the environment to tell the cell to divide. So as you know, cancer cells divide a lot. And when you have amplification of the gene, you have too many copies of it. So you have too many receptors lined up on the surface of the cell that are all capable of sending that signal. And so you tend to get cell proliferation. And what the targeted drug, trastuzumab, does is shut down that activity of that protein receptor. And so finding out who's going to respond to that drug depends on knowing that there's too many copies of that HER2 gene there. So that's the reason why we do the testing, is to find those patients who would be predicted to respond best to the drug. The second test here is microsatellite instability status, and basically that is another way to predict if patients are more likely to respond to therapy, but in this case it's immunotherapy. So I'll show you in a couple minutes a slide about the pathogenesis there of why microsatellite instability is thought to be linked to response to immunotherapy drugs. So first of all, let's get back to HER2, and then we'll deal with microsatellite instability. So with HER2, there's basically a common test that's done, which would be an immunostain, and if that protein-based test is equivocal, then we go on to look for activation of the gene by other mechanisms. And what I'm showing you here is that in stomach cancer, about 20% of patients will have an activated HER2 gene. It's commonly activated by gene amplification, which means too many copies of the gene. But there is an alternate way that it can be activated that doesn't necessarily result in protein overexpression, and that would be a mutation of the gene that just constitutively tells that protein to send the signal to the cell to divide. And interestingly, there's really two diseases where the gene tests are done. One is stomach cancer and the other is breast cancer. And both of those situations will help figure out which of those cancer patients are more likely to respond. And since trastuzumab is a drug that's available worldwide, it's on the WHO list of essential drugs, so even low-resource countries tend to have that drug. It's a really important test worldwide for the approximately almost a million patients who per year that get stomach cancer. But getting back to the mechanism here, so what I'm showing you in the upper right is the kind of test result that I would look through in the microscope. You can see three big purple blobs, and those are 3 cells in a row looked at through a microscope. The cell on the left is a normal cell. It has only 2 red dots in it, each one corresponding to the normal copies of the HER2 gene. That gene is present in 2 copies because everybody has 2 copies of the gene in every cell in their body. One was inherited from their mom, the other was inherited from their dad. The other 2 cells, the 2 cells on the right, have way more than 2 red dots in the cell, and that's HER2 gene amplification. Those are cancer cells. So in gastric cancer, when we see this pattern, we say, oh, this patient's HER2 amplified. They would be more likely to respond to this trastuzumab drug. Interestingly, there is this other mechanism then for activation of HER2, which is point mutation. And those missense mutations are even more common in gastric cancer than they are in breast cancer. And we don't know as much about them, but in clinical trials, there's a lot of sequencing of the entire gene here. And what I show you in the bottom right is sort of a depiction of the different domains of the HER2 gene, and the little dots above the gene are where different patients have had activating mutations in that gene. And we think that those patients are more likely to respond to Herceptin, but we still only have anecdotal data on that point, and so enrollment in clinical trials is really essential to answer the question about which mutations are really correlated with drug response. So we're still awaiting more data on that, but this is basically what pathologists do, is look at these genes and try to identify some things that will help to sort out patient response predictions. So then getting to the second gene test that's standard of care, at least in advanced or metastatic cancer patients, that's the microsatellite instability status. So it characterizes a class of about 20% of gastric cancers, And what happens, we think, and we don't know why this happens, but there's a gene called MLH1. That gene gets turned off. It normally should be expressed, but it's turned off because the promoter of it becomes methylated. And when that happens, you don't make the protein encoded by that gene. Well, that protein is essential for repairing defects in DNA. We think people get mutations all the time, but normally they can actually repair and correct those mutations. But without this protein, they can't do that. Well, that leads to an accumulation of mutations in these cancer cells, which then, because some of those mutations are occurring in protein-coding genes, is producing new proteins that the body's never seen before because they're coming off of a mutated gene. Those new proteins then, called neoantigens, are what's not being recognized well by the immune system, but we think might be why immunotherapy works so well in patients who have this microsatellite instability problem, is because basically they're making these foreign proteins that are mutated, and if only we could trigger their immune system to be more active with the immunotherapy drugs, then the immune system will fight off the cancer on its own. So it's basically just triggering a little bit more of a natural immune response to the cancer cells. And it is specific then for cancer and not for normal cells that wouldn't have the MLH1 promoter methylated. In terms of what we do in the laboratory, there's basically 4 different laboratory tests we can use to identify this microsatellite instability status. The 2 cheapest ones are listed on the top, and by far MLH1 immunostain is the more common of all the tests listed here because it's cheap and it's histology based, so it's very accurate. MLH1 methylation is another option, and then also looking for microsatellite instability, which was sort of the first one discovered, which is why we call that the name for this class of cancers, but basically it has to do with just the fact that there's certain parts of your genome that should be in a certain configuration, but they're in an unstable configuration. And so looking at the length of those unstable regions is what we do in the laboratory. And then finally, there's a really expensive test, but it's incredibly powerful, and that's a next-generation sequencing assay, looking at the sequence of many different genes, including ERB2 and HER2 and many other genes. And it can also, at the same time, look for microsatellite instability and look for what we call tumor mutation burden, which is a marker of how many mutations are there, which really characterizes this. All of these tests really characterize this class of 20% of gastric cancers that have a different response to immunotherapy and therefore need to be identified separate from all the other cancers that wouldn't be predicted to respond as well to immunotherapy. So those are the 2 main tests that we use routinely in gastric cancer, especially the gene tests we use in advanced cancers. Now I wanted to move into a little bit more of what's coming down the pike and what we know about the genetics of gastric cancer that might really help us to devise new therapies and new ways to predict which patients are going to respond best or which patients might consider one clinical trial over another trial. And what I wanted to highlight here is a study that was published about 4 years ago, where the Cancer Genome Atlas group basically sequenced and did extensive genomic testing of 300 or so gastric cancers. And what they found is there's really not just one disease going on here. There's 4 separate diseases when you really look closely at the molecular basis of what's causing the disease. And so let's look briefly at the 4 different kinds of cancer. So we've just mentioned the microsatellite instability group, which is about 20% of all cancers and predicts response to immunotherapy. The second type I want to emphasize is the Epstein-Barr group. It's about 10% of cancers. And don't be alarmed if this virus sounds familiar to you. It's the cause of what we could call infectious mononucleosis, a very common disorder in adolescents. And virtually everybody walking down the hallway has had Epstein-Barr virus infection at some point in childhood or adolescence. And so this virus alone is certainly not causing cancer, but it is thought to be involved in this unique form of cancer because it's present within the malignant cells. And there's a very unique gene mutation pattern in these types of cancer to where there's a PIK3CA mutation or JAK2 amplification, both of those potentially treatable with clinical trial experimental drugs, and also hypermethylation, again, affecting gene expression and potentially treatable. And there's a lot of case report data coming out now suggesting response to immunotherapy in this group as well, because after all, they have a foreign organism within the malignant cells. They're making potentially foreign proteins that would understandably potentially be overcome with something that triggered the immune system to fight a little bit harder. The next type of cancer I want to mention is actually half of all gastric cancers. It's called the Chromosome Instability group. It has a p53 mutation that causes then a failure to trigger cell death when mutations or gene amplifications are present. So you get a lot of gene amplifications, including many in genes like EGFR, for which we have targeted drugs. And so there's a lot of work going on to try and match up targeted drugs with which particular gene amplifications or overexpressions are present. So that's that group of cancers. The last group I wanted to mention is the genomically stable group, which tends to have defects in adhesion between two adjacent cells. And so basically, lining your stomach, you've got a whole series of cells. And if there's a defect that causes them not to stick together properly, then you can have basically a lot of leakage across the lining of your stomach. And we think that because of these adhesion defects that involve the CDH1, RhoA, or RGAP genes, that in 20% or so of cancer patients, this is the mechanism by which the cancer is driven, and also potentially, and maybe Richard Hodge, our next speaker, will tell us more about the pathways and the potential gene therapies that might be available for that group. That group also has something unique, and that is through the microscope, you can see the histologic features of that kind of cancer. It's called the diffuse histology of cancer, and the tumor cells are like individually sprinkled through the tissue, again, because they're not sticking together properly. And so they're not forming the kind of glands that you see in the more typical intestinal histology of gastric cancer. There's also two other histologies we recognize through the microscope that have a lot of lymphocytes, and that includes the two histologies where we're predicting immunotherapy might work the best, because immunotherapy might just trigger those T lymphocytes to fight the tumor cells a little bit stronger. In the Epstein-Barr virus positive group, for which you can see the in situ hybridization result showing the tumor and the virus co-localized. And then the microsatellite instability type that we just mentioned is already FDA approved for response to immunotherapy. The last thing I wanted to cover is monitoring cancer therapy, because there's nothing worse than having a treatment and it not working, but you don't know it's not working. So how do we monitor patients best? Well, what I'm showing you here is the tumor cells in the upper corner there in sort of blue and gray, and those tumor cells occasionally will die and turn over, and And when they do, they release their DNA into the circulation. Likewise, metastases can do the same thing. And so by looking at the DNA in the bloodstream, we can actually see tumor-derived DNA that gives us a clue that tumor is still present in that patient's body. And so at our institution, we have the University of North Carolina Plasma Mutation Panel that's available on a clinical trial basis, where we're monitoring tumor burden to see if patients are responding to therapy by measuring their own tumor markers that we have identified by another sequencing assay, DNA sequencing assay. And we can help determine if the tumor burden is going down over time during therapy. And furthermore, we can try to predict if the tumor is going to recur before the patient becomes clinically symptomatic again. And so that's just one of the uses of this test. And another use is to try and predict drug treatment based on which genes are mutated, or to look at later time points and see if there's a drug resistance mutation. And then the final application would be in distinguishing heritable or germline mutations from those that are the more common acquired ones in tumor cells. So the last topic I want to talk about is cancer prevention. And there's a lot of work going on to try and identify cancers early. And one of the ways to do that is to know if somebody's at high risk for cancer, such as they've inherited genes that run in their family that might predispose them to cancer. And we think maybe about 5 or 10% of gastric cancer patients have a hereditary component to their tumor. And these are just a long list of the different syndromes that have been identified and the genes that are there that can be tested for in clinical grade reportable ways. So if you have cancer at a young age, or you have a lot of family members who are affected by cancers of various types, then you might want to consider asking your physician if you qualify for some sort of testing for a heritable predisposition, because it affects not only the cancer patient's risk of getting a second cancer, but also their family members' risk of getting cancer. And there are some surveillance mechanisms that are in place that are described further in this practice guideline cited at the bottom of the slide. So I think what I've told you today is some of the potential utility of gene tests in gastric cancer, both in terms of what we just mentioned with screening and early detection, and also predicting response to therapy and monitoring tumor patients during their treatment. So just in summary, I think that we have 2 gene tests that we use routinely in gastric cancer, and we have some really novel tests that are being used in clinical trials that target not only human genes, but also viral and bacterial pathogens that are tightly linked to certain aspects of gastric cancer and make it a unique kind of cancer from that regard. And that we can also help monitor patients to see the efficacy of their therapy and perhaps provide some valuable data beyond what radiographic measures tend to do. So I want to thank the Foundation for inviting me today and my colleagues at UNC.
Thank you. Do we have any questions from the audience?
Not sure if this is your area or not, but I've always wanted to know if you are able to do pathology testing on a specimen. Are you able to test a drug on that specimen to see if it will work prior to the patient getting the treatment?
So there's some really innovative, but not, I would say, standard of care ways of taking a patient's tumor specimen and growing it in the laboratory, either in like a plate of tissue culture or actually putting the human tumor cells into a mouse who's been immunosuppressed and then treating that mouse with drug or treating that plate of tumor cells with drug and looking at the response. So there is some work on that, and it is ongoing at my university as well as several other major academic centers to try and better predict, but I wouldn't say that that's standard of care at this point for any particular use.
One of the questions that came in during registration, our older son was diagnosed with adenocarcinoma of the stomach at 17. When should we insist that our younger son get genetic testing and start having lab tests done?
So 17 is awfully young to have cancer, and I would say that going to a genetic counselor is the number one best thing to do in that situation. You need to understand the implications for the 17-year-old, as well as for not just the brother or sibling, but for all the family members. And so talking to a genetic counselor about the options, and there are many options for doing laboratory tests to try and identify if there is a familial predisposition, and also to talk to family members about their options as well.
Any other questions?
Yeah.
I think we have time for one more.
If this is too medically, politically involved, you don't need to answer it. Getting your family genetically tested, how is that going to impact, or does it, or should it impact their health insurance in the future?
Well, luckily, many advocacy groups, and I'm very grateful for the advocacy that this foundation has done, many advocacy groups worked very hard about 10 years ago in the U.S. to pass a law called GINA. It's the Genetic Non-Discrimination Act. And basically, certain forms of insurance are now prohibited from discriminating against people based on the genes they were born with. Because if there's one thing you can't control, it's the genes you were born with. And so I think there are some protections already, but those protections are not universal. And so I think that, it is a political issue, and I think it's something that, we hope that there will be more conversations about and more advocacy to try and get what people really need in terms of, better safety for themselves and better insurance coverage for their families.